Welcome friends, welcome to Science CB. Today, let us discuss about the history of microbiology. As microbes are invisible to the unaided eye, definitive knowledge about them had to await the development of the microscope. The credit for having first observed and reported bacteria belongs to Antony von Leeuwenhoek, a drafter in Delft, Holland, whose hobby was grinding lenses and observing diverse materials through them. In fact, even before the microbial cause of infection had been established, Ignaz Semmelweis in Vienna had independently concluded that puerperal sepsis was contagious. He also identified its mode of transmission by doctors and medical students attending on women in labor in the hospital and had prevented it by the simple measure of washing his hands in an antiseptic solution, for which service to medicine and humanity he was persecuted by medical orthodoxy and driven insane. The development of microbiology as a scientific discipline dates from Louis Pasteur. He introduced techniques of sterilization and developed the steam sterilizer, hot air oven, and autoclave. He also established the differing growth needs of different bacteria and contributed to the knowledge of anthrax, chicken cholera, and hydrophobia. An accidental observation that chicken cholera bacillus cultures left on the bench for several weeks lost their pathogenic property but retained their ability to protect the birds against subsequent infection by them, led to the discovery of the process of attenuation and the development of live vaccines. He attenuated culture of the anthrax bacillus by incubation at high temperature up to 40 to 43 degrees Celsius and proved that the inoculation of such cultures in animals induced specific protection against anthrax. It was Pasteur who coined the term vaccine for such prophylactic preparations to commemorate the first of such preparations, namely cowpox, employed by Edward Jenner for protection against smallpox. The greatest impact on medicine was made by Pasteur's development of a vaccine for hydrophobia. An immediate application of Pasteur's work was the introduction of antiseptic techniques in surgery by Joseph Lister in 1867, affecting a pronounced drop in mortality and morbidity due to surgical sepsis. Lister's antiseptic surgery involving the use of carbolic acid was a milestone in the evolution of surgical practice from the era of laudable pus to modern aseptic techniques. While Pasteur in France laid the foundation of microbiology, Robert Koch in Germany perfected bacteriological techniques during his studies on the culture and life cycle of the anthrax bacillus in 1876. He introduced staining techniques and methods of obtaining bacteria in pure culture using solid media. He discovered the bacillus of tuberculosis in 1882 and the cholera vibrio in 1883. Rouse and Yersin in 1888 identified a new mechanism of pathogenesis when they discovered the diphtheria toxin. Similar toxins were identified in tetanus and some other bacteria. The toxins were found to be specifically neutralized by their antitoxins. Paul Ehrlich, who studied toxins and antitoxins in quantitative terms, laid the foundations of biological standardization. Ernest Ruska in 1934 developed the electron microscope, enabling visualization of the microscopes now we are calling viruses. The development of tissue culture techniques has permitted the cultivation of viruses. The study of immunity had to await advances in protein chemistry. The pioneering work of Karl Landsteiner laid the foundations of immunochemistry. In 1955, Niels Stern proposed the natural selection theory of antibody synthesis which attempted to explain the chemical specificity and biological basis of antibody synthesis, signifying a return to the original views of antibody formation proposed by Ehrlich in 1898. Frank Burnett in 1957 modified this into the clonal selection theory, a concept in which, with minor alterations, holds sway even now. The last few decades have witnessed an explosion of conceptual and technical advances in immunology. Alexander Fleming in 1929 made the accidental discovery that the fungus Penicillium produces a substance that destroys Staphylococci. Work on this at Oxford by Florey, Chain and their team during the Second World War led to the isolation of the active substance penicillin and its subsequent mass production. This was the beginning of the antibiotic era. Other similar antibiotics were discovered in rapid succession with the sudden availability of a wide range of antibiotics with potent antimicrobial activity. It was hoped that bacterial infection would be controlled within a short period, but soon the development of drug-resistant bacteria presented serious difficulties. These are the pioneered works which was done by scientists in the history of microbiology. Let us meet in the next video. Every day, new knowledge.